everyone. Welcome to episode 114 of Cell and Gene, the podcast. I'm your host, Erin Harris, and today's episode is one that's going to really open your eyes to where allogeneic cell therapy is headed, specifically outside of oncology. We're diving into the world of allogeneic cell therapy with Brian Culley, CEO of Lineage Cell Therapeutics. Brian and his team are working on something incredibly exciting. They're using allogeneic off-the-shelf cells to repair damage from spinal cord injuries. Now think about that for a second. They're replacing lost or damaged cells in the spinal cord with healthy ones that are manufactured in the lab. Brian walks us through how Lineage's approach works, what makes their OPC1 program different, and how they've designed a new delivery system that's safer, easier to use, and could dramatically improve patient access. We also talk about what it really takes to make allogeneic cell therapy scalable from the perspectives of manufacturing and formulation and clinical delivery. If you've been following the evolution of allogeneic cell therapies, this is an episode you'll definitely want to hear. It's science, engineering, and patient impact all wrapped up into one conversation. And so here it is. Here's my chat with Lineage Cell Therapeutics, Brian Culley. Enjoy. Hey, Brian, welcome to Cell and Gene, the podcast. Thanks for being here.
Thanks, Erin. It's my pleasure.
So I'm really glad that we have the chance to talk again. You and I talked years ago on the Business of Biotech podcast, which was at the time hosted by my colleague, Matt Pillar. It has since been taken over by my colleague and equally wonderful podcast host and chief editor, Ben Comer. I'm getting Matt and Ben confused. So thank you for your time. It's great to have you on Cell and Gene, the podcast, because when we first talked, Cell and Gene, the podcast didn't exist yet. So I'm happy to have you on our podcast. Tell us a little bit about yourself and Lineage Cell Therapeutics and just a little bit about the company's mission.
Well, great. Thanks, Erin. Originally from Boston, so when I hear Matt and Ben, I think of two of the famous actors from there. But
I mean, who wouldn't? Yeah,
exactly. But for the last 7 years, I've been the CEO of Lineage Cell Therapeutics. Lineage is a publicly traded company, so just a note for listeners that they can review the risks of investing in Lineage through the filings we make at sec.gov. And what we focus on is an area of cell therapy that we think is poised for explosive growth, and that's outside of oncology. A lot of what many of your listeners and yourself and even me think about is autologous CAR T because it's revolutionized cancer, and that's fantastic. But there are some really exciting opportunities in non-cancer indications, and what we have done is we have built a portfolio of assets that are all allogeneic. Not just allogeneic, but also seeking to be off the shelf. So truly addressing the cost issue with cell therapy. And in short, these are replacement parts. There are indications and conditions in the world where if you can manufacture in the lab the cell type that is lost, and you can deliver that cell to a patient, it's been shown that in some cases you can bring back the functional recovery that was lost. And that is really the mission of Lineage, is to bring forth this very exciting new branch of medicine.
Wonderful, wonderful. All right, I want to talk a little bit about, before we really jump into exactly what Lineage is working on, the DOST study, all of that. Tee this conversation up for our listeners. Bring us up to speed on the current landscape of spinal cord injury. Challenges facing patients, unmet needs, where are the areas for opportunities? Just talk us through that a little bit.
Yeah, spinal cord injury is amazing. It was one of the sort of first and best ideas for a cell transplant approach. So again, this is making replacement cells, in this case, cells of the spinal cord, and transplanting them to the patient, because of course, after a spinal cord injury, those damaged cells are either destroyed or they're dysfunctional. So it's actually our second most advanced program. The most advanced program, which we partnered with Roche and Genentech, is in a form of blindness. The next most advanced program in spinal cord injury in our hands is relying on oligodendrocyte progenitor cells. And we manufacture oligodendrocyte progenitor cells, again, allogeneic, off the shelf. And we manufacture those cells in order to deliver them to a patient who suffered from an injury. Because the oligodendrocyte progenitor, it's the cell that makes the oligodendrocyte, and the function of the oligodendrocyte is to make the myelin, and that's what's wrapped around those neurons that are in the spinal cord. So if you want to have if you want to have an electrical current that is not chaotic, that's sensible and leads to coordinated mobility, your neurons can't just be present. They need to be wrapped in the myelin, which is the conductive sheath for those neurons. So our approach is to make many, many millions of oligodendrocyte progenitor cells, deliver them right to the target lesion, so they get delivered right to the area of injury of the spinal cord. And if they can wrap those neurons in the myelin that had been destroyed, they may be able to help people gain mobility following one of these spinal cord injuries of any type.
Good. Okay. I want to talk a little bit about the DOST study. So first of all, congratulations on treating the first chronic spinal cord injury participant. Talk us through a little bit of that. And then also, what is OPC1, the broader vision for it? Back in 2022, I believe it was, you and I worked on an article together where at the time you were talking to our Cell and Genes audience about OPC1. A lot has happened since then. So tell us about the DOST study. What does that stand for and where we are with OPC1?
Yeah, so OPC1 was actually developed by other sponsors. It was originally developed by Geron Corporation. At the time, it was one of the most pioneering cell transplant approaches with this replace and restore technology. Prior sponsors treated 30 individuals with spinal cord injury and saw this really evocative phase 1 data, i.e., it looked like people were increasing their mobility after an injury beyond what you would expect. So really exciting. But the program, like many early programs, had some flaws. The manufacturing was not commercially viable in its current form, and even the delivery system was not viable in its form. So we've been working on some of these obstacles to really getting this therapy to the bedside. And one of those obstacles around delivery is that the old way of delivering the cells required this big scaffold that sort of sat above the bed, and it held onto the needle and apparatus, and it was really frustrating because you had to literally disconnect the patient from the respirator. They had to stop breathing so that they were immobile in order to fully position the needle and deliver the cell. So, you've got to go quickly, and that's inadvisable when you're doing surgery on the spinal cord. So what we're doing in the DOST study is we are deploying and testing the performance and utility and safety of this little device. It's barely the size of a loaf of bread, and it attaches directly to the patient's back. So the incision is made, the whole apparatus is attached to the patient. So now imagine the patient is breathing, the chest cavity is going up and down along with the device, along with the flow path of the cells. Now you're able to deliver the cells, take 4 or 5 minutes. You can go a lot slower. So it's an easy, convenient, easier to use, potentially safer way of delivering the active therapy to the patient. And it's one that is not only much more easily deployed over a large number of clinical sites, but also it's been shown to be compatible with a new formulation that we developed. We have a ready-to-use thaw-and-inject formulation. So again, when you think about traditional or classic cell therapy, especially in oncology, you're thinking about all this handling and plating cells and manipulation, maybe gene editing. None of that's applicable. Our cells are manufactured, and when they're delivered to the patient, they're thawed in 5 minutes and they just go right in the patient directly. You don't have all those cleansing and counting steps that other cell therapies have as encumbrances or risks. So it's an important but small trial in just 6 to 10 patients. And you asked about chronic versus subacute. This was really exciting for us. It was it has been heretofore our approach to work on subacute injuries. So these are people whose injuries are just, let's say, 6 to 3 weeks earlier, right? So there's a little bit of time for the inflammation to go down, the wound to settle, and that's when we would deliver our material. But the FDA actually suggested to us that we also try this device in patients who had a chronic injury, which we define as 1 to 5 years old. Why is that important? It's because people who have had a chronic injury generally have plateaued with their performance. So if I had an injury 3 years ago, I'm probably not gaining anything anymore. I have done all of my physical therapy, but I probably have a level that can be considered a baseline. So now if I go and I receive these cells, if I do see a change, that's really exciting. Where did that change come from? So even though it's a small number, just 6 to 10 patients will be treated in this. And the point of this study is to show the safety and performance of this device. Along the way, we're going to treat 3 to 5 people with chronic injuries. We're going to follow them long term for functional changes. And we're going to find out, is anything different after getting the cells? That would be really exciting to know.
Certainly. That's incredible. That's a really, really fascinating innovation. Now, just out of curiosity, is there a specific age range of patient? Is it all anyone is eligible for this procedure?
One of the things that we find really compelling about what we're doing with cell transplantation is that, unlike gene therapy where you're really addressing one problem, so one pathway, right? That's kind of the limitation, is you have to find the patient with the problem and then try and fix it. I sometimes joke that cell therapy is like doing 30,000 simultaneous gene therapies. And the point of that is to say that you really can have a much larger addressable market, because it doesn't much matter to us if the patient is, 16, in their mountain bike crash, or they're 60 from an automotive crash. It doesn't much matter to us if it was an injury that came from some sort of, diving versus, some people, even just routine procedures can sometimes lead to spinal cord injury. So what we have is we have a very broad opportunity to replace this cell type in a large patient population. Now, we're not, obviously leading with pediatrics. We do follow all of the traditional, good rules of thumb of clinical development. But I think it's important for listeners to appreciate that when you are identifying a specific cell type, whether it's spinal cord cells or the islet cells for diabetes that could be replaced and show to be curative, or in Parkinson's disease, when you're thinking about replacing dopaminergic neurons, there are specific cell types that if you can put them in a patient, you probably can engender a response, provided that they're durable and provided that they're functional and they remain integrated in that patient. And in most cases, we assume that this is a one and done procedure, and that is the vision for OPC1 for spinal cord injury.
Fascinating. Excellent. I want to talk a little bit about the delivery device itself. So maybe take us back a little bit more. What prompted the development of it, and what technical or clinical hurdles, is it really designed to overcome and is overcoming?
Yeah, it's a great question, because it's actually in many ways a point of frustration for our company. Okay, so what do I mean by that? Is that cell therapy and maybe cell transplantation obviously has led to some really incredible clinical outcomes, often in relatively small patients. I mean, there are 4 companies out there, we're one of them, that have shown that you can increase vision by delivering retinal cells to patients with dry age-related macular degeneration. But in the aggregate, the public data, there's only like 24 people that have ever had this reported. Now, those are 24 people that are doing something that never happens naturally, right? Nobody ever nobody ever heals automatically from that condition. But that is really the point, is that there's really exciting clinical data, and sometimes the commercially necessary aspects of a therapeutic are not catching up, right? So whether that's manufacturing, having a line on site onto control or reproducibility, purity, potency, or delivery, which I think is an area that's often overlooked. Because again, when you think of oncology, you usually just kind of firing the cells into the circulatory system, whereas with specific niches in transplantation, you're sending the cells off into a very discrete compartment or area. So what we saw with the OPC1 program was a necessity to develop an improved delivery system because the clumsy old scaffold, give you an example. Many beds in different hospitals are different sizes. So if you develop a scaffold system to hold a needle in a precision way, and then you want to open up a second site and they bought their beds from some other vendor, oh my
Sure.
gosh, like now you got to go develop a whole new system, right? And it's got 100 parts and they got to be autoclaved and counted and assembled. And it's a pain in the butt.
Yeah.
And that's not a product, right? So the inspiration originally was if we wanted to run a larger trial, we need an easy to use system. It's not about the activity of the cells. It's about making sure from beginning to end, the totality of the program exhibits things that are consistent with commercial viability and commercial success. It's not exciting for investors. It's not sexy, but it's necessary. And
Hmm.
so the trial that we're doing is about developing a device that, yes, can be deployed to many sites, is compatible with our immediate use thaw-and-inject formulation. It's a very straightforward XYZ manipulator that we think will be safer because you don't have to disconnect the kid from the respirator. So it really solves multiple problems for us while not really adding a lot of risk, right? A small study. And in the meantime, we're working on the design of larger studies and understanding our product. And in fact, one of the things we've done is we've revisited how the cells are manufactured. So we have a much better way, a much cleaner way of developing the cells. So we think we have a better product, better delivery, better formulation. These are all the things that help you at the end of the marathon, not, who cares who's winning at halfway. And so that's why it's important for us to have this device data.
Excellent. Okay, thank you. Now, you said earlier that OPC1 is an allogeneic off-the-shelf transplant composed of oligodendrocyte progenitor cells. Excuse me. Did I get that right?
Yes, excellent.
Okay. um, so, talk to us a little bit about what makes these cells particularly suited for spinal cord repair and, potentially restore function for patients.
Yeah, there are a lot of approaches you might imagine. Previously people have used undifferentiated stem cells in a lot of different settings. I remain unconvinced that undifferentiated stem cells have much benefit in settings like spinal cord injury. But if you deliver the cells that are needed, you replace the ones that have been destroyed, then I think you can start talking about, functional recovery, i.e. mobility. And so when you think about the different cell types, because there's so many different cells that do comprise the spinal cord, you don't really lean necessarily toward astrocytes, for example. Astrocytes don't make myelin, but we know that myelin is necessary. Same with neurons. There are a lot of different neuronal types. So what we do is and this comes from years of preclinical work. The oligodendrocyte progenitor, and it's important to say progenitor because it allows you to get a little bit of diversity in your final product. But the oligodendrocyte progenitor giving rise to a population of oligodendrocytes that presumably all of which can manufacture myelin, that insulation that I talked about previously. That's really what is important here. It is a distribution, to be clear, we do characterize our material extensively, right? You can use things like bioinformatics and RNA-seq technologies that barely existed, not too long ago, but now are available to really understand your material. And so, for example, and this sounds a little shocking, but I think it's also highlights our capability. The original clinical material had a population of epithelial cells. Now, that's not what you want going into the spinal cord. So in changing how we manufacture the cells, we were able to control the differentiation to eliminate that population of cells. And you can show that data to FDA or to investors, and it's really compelling to say, hey, I understand my process. I can make the target cell. And what's great about cell transplantation compared to, say, small molecule drug discovery, which I did early in my career, you don't have to worry about target validation. We know what the target is. It could be the auditory neuron or the RPE cell or the oligodendrocyte. The many hundreds of thousands of years of evolution millions of years have given us the information about what we need to make. The challenge is the recipe. Can you make it? Can you make it affordably? Can you make it
Mm,
reproducibility? Because if you do put it in there and it's stable and engrafted and doesn't cause problems, it'll probably work.
perfect. Um, I want to talk a little bit more about patient access for one. Everything we're doing here is for patient access and how to improve patient access. So when we're talking about the immediate use thaw and inject formulation for OPC1 and delivery system that we talked about avoids that stopping patient ventilation during administration, how is that ultimately impacting real world patient access? Because that's a drastic change from, like you said, from the scaffolding where that whole process has, where it's progressed to now. What does that really look like for patient access?
Yeah, I think, you know, we don't limit ourselves to just the excitement of clinical benefit. You know, if the patient never receives the therapy, you know, your work is questionable, right? And all of us are, you know, committed very deeply to these journeys. So we really try to develop products that from start to finish have the, you know, the safety, the delivery, the efficacy, all of the characteristics that you need. One portion of that around the device is sort of physician pointing, right? How do we make it easier for physicians? But there's also a practical side. You know, if we have to run a larger trial to answer the question, how well does our therapy work? Um, Trying to run a larger trial at a very limited number of sites, you know, it'll take forever. It's just incompatible with the realities that you need capital to do these studies. So you have to be investing in every aspect of your program. It's not one of these, you know, make a better mousetrap, the world will beat a path to your door. You have to make a mousetrap and then walk around door to door and explain why it's better. But we've had a, you know, good fortune with that. I mean, this is a program that's previously supported by the California Institute for Regenerative Medicine. We have another grant in front of that same group to see if we can get some additional support. But you know, in thinking about patient access, one of the things that we do, and you know, I hope a lot of companies do this, but we go and we talk to the patients. You know, I've
Mm-hmm.
spent time around the country at the homes of people with spinal cord injuries and learned about how they have all sorts of different equipment and toys and the diversity of injury and you know, learning things like, you know, just a little bit of rotation of the hand that doesn't look like very much, but that's the same movement that allows you to rotate a wheelchair 360 degrees. So really small changes can lead to very important quality of life improvements. And you really have to be working hand in glove with the patient in order to understand what matters to them.
Sure. Makes perfect sense. Finally, I want to talk a little bit about the annual Spinal Cord Injury Investor Symposium. You, Lineage, co founded it with the Christopher and Dana Reeve Foundation. Talk to us a little bit about the symposium and how the ongoing effort is shaping spinal cord injury research.
Yeah, this is such a we really love this program. So, you know, anyone who thinks of spinal cord injury, you know, I think the number one name
Sure.
is the Reeve Foundation. So, you know, everyone, well, maybe less so nowadays, but you know, we think of Superman
Mm-hmm.
because, of course, Chris Reeve, you know, suffered from a tragic and very severe spinal cord injury. So we want to work with you know, the best and the brightest. So we partnered to create this symposium for a number of reasons. You know, this is a very complex disease. It may not have a single solution. You know, we may need to investigate all sorts of partnerships and collaborations of different forms. There aren't enough of us working in the space, so we see an education. You know, investor education helps stimulate an interest in the space. We can collaborate with, you know, ostensibly competitors. You know, I'm really happy that OPC1 probably could work very well with, you know, eSTIM technologies or brain chip interfaces or you know, small molecules, you know, growing neurites. You know, it probably is compatible with all that. But even if it weren't, you know, we're just really strong believers that we're trying to improve the condition. And you know, sometimes you have to work with others. So that program, you know, we get insights from the people who go there. It helps us with our brand and our reputation, showing that we're committed to this field. Networking, we get to the very best, surgeons and researchers in the field, and really just trying to have a leadership position. It's a spinal cord injury is different. I'll kind of wrap with this. If somebody smokes 4 packs of cigarettes a day and develops lung cancer, you kind of saw that coming.
Mm-hmm.
Nobody expects to have a spinal cord injury.
Sure.
And I get the phone calls from time to time, from the dads saying, oh, my kid used to, whatever, play soccer, play baseball, did this, did that, and then he had an injury. Can you help him? Because it tends to be young people, and it tends to be males. And they're incredibly sad because they've had their whole life torn away. So we have an obligation, we have a duty to really do the right thing here. And if that means, we're helping some other competitors along the way, that's okay, because we should all be working on the main goal, which is to improve mobility, function, quality of life for the caregivers, the docs, and of course the patients.
Sure. I love that, with the patient focus always as the primary focus. I love that. Well, listeners, that takes us to the end of this episode featuring Brian Culley, CEO of Lineage Cell Therapeutics. Brian, thank you so much for your time today. This was incredibly insightful, and I'm so glad to finally have had you on Cell and Gene, the podcast, and to bring our listeners up to speed on exactly what Lineage is doing for SCI. So thank you.
It's been my pleasure, Erin. Thank you as well.
And thank you to listeners for tuning in, and we will talk to you soon.